Question number 23. It is based on replication. This question is based on replication of DNA. What is replication? Replication, that is, see this DNA molecule and its base sequence is A, T, G, C at one strand, and the complementation principle, according to complementation principle, T, A, C, G on other strand, and these are joined together by means of hydrogen bond. When DNA replicates, see this one, gradual separation of these two strands A, T, G, C, T, A, C, G, followed by the synthesis of new strand, that is, this strand required complementary strand, so new strand is synthesized by this one, and similarly new strand is synthesized by this one. T, A, C, G, A, T, C, G, hydrogen bond is there. See the parental DNA molecule, this is parental DNA molecule, this is parental DNA molecule, and see the newly formed DNA molecule. See the newly formed DNA molecule. This is parental DNA molecule and this is the daughter DNA. This is parental DNA, this is daughter DNA molecule. See the sequence of bases in that DNA and see the sequence of bases in newly obtained DNA molecule. The newly formed DNA molecules are exact copy of the parental DNA. Newly formed DNA molecules are the exact copy of the parental DNA. Exact copy is known as replica. Exact copy is known as replica, and that's why the method of replication, method of DNA formation is known as replication. When DNA synthesizes DNA, it forms its own exact copy, that is called replica, and the process is known as replication. This is the meaning of replication. This is the meaning of replication. Then see, these two DNA molecules are obtained from this one, and you see this strand is now in DNA molecule, and from this side this strand is from the parental DNA molecule. Only one strand is synthesized. Out of two strand, only one is synthesized in every DNA molecule. In every DNA molecule, one strand is contributed by the parent, whereas the other strand, that is complementary strand, is synthesized by the cell itself during the replication. So method of replication is semi-conservative. This is second question. This is second important point. That is, method of replication of DNA is semi-conservative. And DNA, third important information, DNA replicates at the time of cell division and S phase of cell cycle is exact answer. S phase of cell cycle. So these are the three important informations regarding the replication of DNA. That is, DNA is able to form its own exact copy by the process of replication. Second, the method of replication is semi-conservative. In every DNA molecule, one strand is parental, other one is new. And third, DNA replication takes place in S phase of cell cycle. Next, fourth important information, that is, the entire process is enzymatic. DNA replication is an enzymatic process. This is an enzymatic process, and enzymes which are involved, the group of enzyme involved in DNA replication, that group is known as replisome. That group is known as a replacome. That group is mentioned below, but the order is not correct. And in this question, we have to arrange these enzymes according to their order of participation, then check the correct sequence. So first of all, this is the double stranded DNA molecule. This is double stranded DNA molecule. First, there is a separation of two strands. There is a separation of two strands. So this indicates the separation, gradual separation of two strands. For this enzyme is needed, that is helicase. So process start with the third one. That is role of helicase. And if it starts with the 3, then there are two options, 1 and 3rd. They start with the 3. And even the second one is also 3rd with. So we have to choose now. We have 3 options from which we have to collect the answer. So first one, that is 3. That is role of helicase for the separation of two strands. Then during the separation, tension is released by that is 4th topoisomerase enzyme. Topoisomerase enzyme releases tension. Now we have only 2 options left, then 1st and 3rd, because here the second one is 1st, that's why this is already gone. So 3 and 4 are now, 2nd and 4th are now cancelled. Now come to the 3rd one. First one, separation of two strands. Second one, release of tension. Third, DNA replication requires RNA as primer. DNA replication requires RNA as primer. So definitely primase activity is there, because RNA primer acts as stepping stone. So according to sequence, 5th one, 3, 4, 5, 3, 4, 5, and this is 3, 4, 5. Almost clear, that is answer 3rd, but let's check further. That is RNA primer, it is responsible for the attachment of a primer, followed by the polymerization. Polymerization takes place with the help of DNA polymerase number 3. Actually, DNA polymerase is a group of enzyme. It involves 1, 2 and 3. And 3 is the main enzyme. DNA polymerase 3 is main enzyme responsible for replication. So 3, 4, 5, 2. 3, 4, 5, 2. According to sequence now, quite clear. Position is now quite clear, that is 3, 4, 5, 2. During the replication, the proofreading work and repairing work is done by 1. That is DNA polymerase 1. It's a proofreader. DNA polymerase 1 is proofreader. Whereas 3 is actual replicating enzyme. Third is actual replicating enzyme that involve in the replication. Then during replication, at one strand, DNA grows in bits. One strand grows continuously, whereas other strand grows in bits. These bits are called Okazaki fragments. And these Okazaki fragments are joined together by means of ligase enzyme. So 6th is the last. In this order, 6th one is last. Now position is absolutely clear. 3rd, 4, 5, 2, 1, 6. So correct order is 3. Answer number 3. Correct order of this question, the participating enzyme, is 3. Now come to the 24th question. Okazaki fragments are formed by one of the strand of DNA that is known as lagging strand. One strand that forms Okazaki fragment is known as lagging strand. That strand is called lagging and other strand is known as leading. At the time of replication, two strands gradually separates. These two are the two strands which gradually separates. Polarity of this strand is 5 and 3, and this polarity of this strand is opposite polarity, that is 3, 5. At the time of replication, the direction of synthesis of new strand is always 5 to 3. This is another question of pre-medical. That is, strand always grows in one direction, that is 5 to 3. So see here, from this strand, the process starts and it grows continuously. This strand grows continuously as is got the open session. This strand grows continuously. The strand on which the DNA grows continuously is leading strand. And here, to follow the 5-3 polarity, so process starts from open areas only. And it is in bits. This is in bits. That is, activity of RNA primer is more on the other strand. Activity of RNA primer is more on this strand. And second one, this strand grows in bits, and these bits are known as Okazaki fragment. These are known as Okazaki fragment, and the strand on which question says the Okazaki strand formed by one of the strand, that is known as lagging strand. That strand is known as lagging. So, strand on which DNA grows in bits is lagging strand. That is known as lagging strand. This is question number 24. 25th, the statement is correct. Bacterial DNA replication. At the time of replication, there is a specific site. There is a specific site from which DNA replication starts. That site is known as Ori site. That site is known as Ori site. So in prokaryotic cell, there is only one Ori site. Only one Ori site. That means 1 and 3rd possible. Second one, the replication of DNA. Replication of DNA takes place in 5 to 3 direction only. 5 to 3 direction. But with regards to the two strand, at one strand it grows in this direction, whereas it grows in this direction on the other strand. So single Ori site and bidirectional process. Single Ori site and bidirectional process. Number of Ori sites are seen in eukaryotic cell. New strands of DNA already mentioned, that is 5 to 3 direction, with the help of enzyme DNA polymerase 3. DNA polymerase number 3. DNA polymerase number 3. This enzyme synthesizes DNA in one direction, that is 5 to 3 direction, and the speed of polymerization is 2000 base pair per second. This is the speed of this enzyme. Transfer of genetic information from DNA to messenger RNA. DNA to mRNA. That is known as transcription. This is called transcription. This is known as transcription. That is, there are two strands of DNA. And the polarity of these two strands 5335, 5335. These two strands join together by means of hydrogen bond. At the time of transcription, At the time of transcription, only one strand acts as template. So, and the template strand's polarity is 3 to 5. Template strand polarity is 3 to 5. The polarity of the template strand is 3 to 5. So this strand is template strand, this strand is template strand, and the direction of the formation of the RNA is 5 to 3. That is 5 to 3. The RNA direction of synthesis of RNA is 5 to 3 with the help of RNA polymerase enzyme. With the help of RNA polymerase enzyme. That is, RNA polymerase enzyme is required for the transcription. This enzyme is also known as transcriptase. This enzyme is also known as transcriptase, or we can say DNA dependent RNA polymerase. DNA dependent RNA polymerase. DNA dependent RNA polymerase. DNA dependent RNA polymerase enzyme, that is known as transcriptase. So these are the two strands. The polarity with 5-3 is coding strand. This strand is coding. This strand, the opposite one, is known as non-coding or template strand. This is non-coding or template strand. The actual strand which synthesizes RNA is called template strand. So this is coding, this is template or non-coding. This is template or non-coding. Because base sequence of RNA resembles with this strand, the only difference that is, instead of thymine, uracil is present in RNA. Instead of thymine, uracil is present in RNA. That's all for 27th question. Come to the 28th. A DNA sequence is read by the RNA polymerase that produce complementary anti-parallel strand is known as. That is called primary transcript. That is known as primary transcript. That is known as primary transcript. I have already mentioned this is DNA molecule double stranded. This is DNA molecule double stranded. Polarity 5-3, polarity 5-3 and 3-5, and from 3-5 strand, that is from this strand 3-5 polarity, RNA polymerase enzyme forms RNA. RNA polymerase enzyme, RNA polymerase enzyme forms RNA molecule. This is RNA molecule formed by this strand, right? And this is primary transcript, complementary anti-parallel RNA strand. See this one. Suppose here A, T, G, C is there. Then in that case, T, A, C, G is an RNA. And when this strand forms RNA, the T, that is complementation rule, is applicable. Here A is there, then AT, but in T, RNA, T is not found, so U is there. Then C, here G is there. Then here G, here C is there. This is the sequence of bases on RNA. Sequence of bases on RNA formed by the 3-5 strand. Formed by the 3-5 strand. But you see, the sequence is very similar to the sequence of 5-3. That is, these codes are actually transferred to the RNA. The difference is that only, that is, thymine is not found. Thymine is not found in RNA. Instead of thymine, uracil is present. So primary transcript is formed. Then binding site of RNA polymerase enzyme. Transcription unit. Transcription unit comprises of three regions. This is DNA molecule, polarity 5335, and this is known as coding strand. This is known as coding strand. And transcription unit comprises of number one, with respect to coding strand. Promoter is present in the beginning, and this promoter is binding site of RNA polymerase enzyme. Then this is the region of a DNA which is transcribed by the RNA polymerase. So this region is known as structural. This portion is called structural gene. This portion is known as a structural gene. Then at the other end, that is towards three end of coding strand, that is three end of the coding strand, this area is known as terminator. This is known as terminator. So with respect to the coding strand, this is promoter region. This is promoter, and promoter is binding site. Promoter is binding site of RNA polymerase. Then this is the area where the replication RNA formation takes place, that known as a structural gene. This is known as a structural gene. This is called structural gene. That is, prior to structural area, promoter is present. Upstream, we can say upstream. Prior to structural, promoter is present, and after the structural, terminator is present. This is terminator, that is responsible for the termination of this process. This is terminator area. And promoter is binding site of RNA polymerase enzyme. Promoter is binding site of RNA polymerase enzyme. This is the binding site for RNA polymerase enzyme, responsible for the replication. Which of the subunit? RNA polymerase enzyme actually comprises of 2 alpha chain, alpha, then beta, beta 1, omega, and 1 unit, that is sigma factor. Sigma factor is the master or key chain, and the remaining portion, that is this one, is core enzyme. This one is core enzyme. I mean to say RNA polymerase enzyme is holo enzyme. Comprises of these chains: alpha, beta, beta 1. This is beta 1. This chain is beta 1. And omega and sigma. Sigma is the key chain. So which of the subunit of RNA polymerase totally required to initiation? Transcription is initiated by the sigma factor and elongation is done by the core enzyme. Transcription initiation is done by the sigma factor. Then core enzyme responsible for the elongation. And termination signals are given by rho factor. Rho factor responsible for the termination. This is the role of different chains in question number 30. The question is asked to initiate transcription, then sigma factor is the correct answer. DNA chain acting as template for mRNA synthesis has following order of bases: A, G, C, double T. Let's check. A, G, C, T, T, DNA chain acting as template. This is template chain. This is template chain. Synthesizes RNA in this direction. That is, RNA is formed in this direction over this chain. So A combines with T, but T is not found in RNA. So here we write U, then C, then G, then A, G, C, and U. Check it, the sequence. That is, I think the last one resembles. Because in RNA, uracil is present, thymine is not there. Uracil is present, thymine is not there. So, principle of complementation applies. One, and second one, in RNA, uracil is present instead of thymine. So, that's why the correct answer is this one, because this is the template strand. Template strand synthesizes RNA molecule, and the synthesis of direction is Y3. In most of the eukaryote, a structural gene is monocystronic. That is responsible for one particular polypeptide chain. The gene which is responsible for the synthesis of one particular polypeptide chain, that is monocystronic. If it can synthesize number of polypeptide chain, that is polycystronic. So monocystronic DNA is found in eukaryote, whereas polycystronic DNA in prokaryote. Polycystronic DNA, it is found in eukaryotic cell. So answer is now very clear. That is, this gene synthesizes only one particular peptide. What is cistron? Cistron is a DNA segment responsible for one polypeptide chain. Cistron is DNA segment responsible for a synthesis of one polypeptide chain. Monocystronic, that is one polypeptide chain is synthesized by the polycystronic DNA, that is DNA segment acts as polycystron, that is ability to synthesize number of polypeptide chain, and applicable for eukaryotic cell.